Blood tests. We have all been there. You sit down in a chair. A nurse ties a rubber band around your arm. You look away as the needle goes in. They draw out what feels like a surprising amount of blood. Vials fill with different colored tops. Each tube is for a different test, for a different machine, for a different analysis. The process has been the gold standard for decades. It works. It gives doctors a detailed snapshot of what is happening inside our bodies. Cholesterol levels, liver function, signs of infection or disease. But it requires a significant amount of blood for each separate diagnostic test. That can be uncomfortable and stressful. After the draw, those vials are carefully labeled, packaged, sent off to a centralized laboratory, sometimes in the same hospital, sometimes hundreds of miles away. Technicians unpack and get to work, running different tests on different machines, a process that can take hours or days. You're left waiting for a phone call, waiting for results in an online portal, waiting for answers about your health. The system runs on logistics, specialized equipment, and manual labor. It's a slow, resource-intensive dance of collection, transportation, and analysis. And it has changed very little over time. That reliance creates barriers. Remote or underserved areas struggle to access clinics. Fear of needles causes real anxiety for many. Some avoid needed tests entirely. Chronic patients face repeated appointments and blood draws. It's accurate, but not designed for convenience, accessibility, or comfort. The amount of blood required is a real issue for some groups. Newborn babies, elderly patients with fragile veins, cancer patients undergoing chemotherapy. Multiple vials can be physically taxing and sometimes risky for them. The traditional method demands a macro sample to answer micro questions. What if we could flip that script? What if the same, or better, answers came from a sample thousands of times smaller? That question has driven scientists and engineers for years, pushing them to rethink diagnostics and search for a better, smaller, faster way. Imagine a world where a comprehensive health checkup doesn't require vials of blood. Instead, it just needs a single tiny drop, one small prick on your finger like the kind people with diabetes use to test their blood sugar every day. Close-up of a single drop on a fingertip. This is the revolutionary idea that has captivated the medical and tech industries for over a decade. Miniaturize the entire laboratory process so that it can work with a minuscule amount of blood. Instead of needing separate, large machines for each test, what if a single compact device could analyze dozens, hundreds, different health markers all at once, all from that one drop? These technologies are the key to making the one-drop dream a reality. A lab on a chip is a small, transparent chip no bigger than a postage stamp. Tiny channels, chambers etched into it. When a drop of blood is placed on the chip, it is guided through these microscopic pathways. Inside the chip, the blood can be separated, mixed with different reagents, analyzed for various biomarkers. Each channel can act like a miniature test tube. Each chamber can be a miniature sensor, like taking a massive, room-sized laboratory and shrinking it down onto a tiny piece of plastic or glass, making diagnostics portable, fast, and incredibly efficient. The potential impact of this shift is monumental. It could completely democratize healthcare. Instead of waiting days for results from a distant lab, a doctor in a small rural clinic could get answers in minutes at the point of care. Faster diagnoses and immediate treatment decisions, life-saving in situations like sepsis, life-saving in a heart attack. For individuals at home, it opens the door to proactive health monitoring. Imagine being able to track your cholesterol, track vitamin levels, or detect early signs of inflammation with a simple home test. Empower people to take control of their health. Move from reactive medicine to a preventative model. However, this big idea comes with equally big challenges. Working with such a tiny drop of blood is incredibly difficult. A single drop contains cells, it contains proteins, it contains other molecules, but the quantities of each are extremely small. This makes it hard to get a consistent and accurate reading. A microscopic speck of dust can throw off results. A tiny air bubble can disrupt the test. Slight variations in temperature can change the reading. The initial dream hit a wall of scientific reality. The hardware was developing, but making sense of the messy, minuscule data from a single drop of blood proved to be a formidable obstacle that technology was not yet ready to overcome. This is where artificial intelligence, or AI, changes the game. While microfluidics provided the physical stage for one-drop testing, AI provides the intelligence needed to interpret the performance. The data generated from a tiny drop of blood is not just small, it's also incredibly noisy and complex. When you're looking for faint signals of a disease, like a few rogue cancer DNA fragments floating in a sea of healthy cells, it's like trying to hear a whisper in the middle of a rock concert. The human eye or traditional statistical methods would miss these subtle clues completely. They would be lost in the background noise of natural biological variations and tiny measurement errors. AI, however, is built for this exact kind of challenge. Machine learning, a type of AI, uses powerful algorithms to sift through massive amounts of data and find patterns that are invisible to humans. Scientists can train an AI model by feeding it thousands of blood sample results from healthy individuals and from patients with specific diseases. The AI learns to associate incredibly subtle combinations of biomarkers with a particular condition. It doesn't just look at one marker in isolation, it analyzes the complex interplay between hundreds of different data points simultaneously. For instance, it might learn that a slight dip in one protein, combined with a tiny increase in another and a specific pattern in a group of cells, is a strong predictor of early-stage kidney disease. This ability to find hidden correlations is what makes AI the perfect partner for microsample diagnostics. It acts as a powerful signal booster and error corrector. If a measurement from one tiny channel on a lab on a chip seems slightly off, the AI can cross-reference it with data from dozens of other measurements to determine if it's a genuine signal or just random noise. This dramatically improves the reliability and accuracy of tests performed on very small samples, overcoming one of the biggest hurdles that held the technology back for years. It's not just about shrinking the lab anymore, it's about adding a super intelligent brain that can make sense of the complex information the miniature lab produces. So the modern approach to one-drop blood testing isn't just about clever hardware. It's a marriage of two groundbreaking technologies: microfluidics to handle the blood and AI to interpret the results. The chip does the physical work of separating and sensing, while the AI does the cognitive work of analysis and diagnosis. This powerful combination is what is finally turning the dream of comprehensive, single-drop testing into a tangible reality. It's the reason why, after years of skepticism and high-profile failures, we are now seeing real, approved products and promising research that could soon change medicine forever. The journey from vials to a single drop has been long, but with AI as our guide, the destination is finally in sight. So, how do we begin to shrink an entire laboratory? The first step is to control the movement of incredibly small amounts of liquid. This is the science of microfluidics. Think of it like a city's plumbing system, but shrunk down to a microscopic scale. Instead of large pipes carrying water, microfluidics uses tiny channels, some even thinner than a human hair, etched onto a small chip made of glass, silicon, or plastic. When a single drop of blood is placed on this chip, it doesn't just sit there as a blob. Instead, it's pulled into these channels by forces like capillary action, the same force that allows a paper towel to soak up a spill. This allows us to precisely guide the blood to different destinations on the chip. Once inside these microscopic highways, the real magic begins. The blood isn't just flowing from one point to another, it's being actively managed. We can create tiny intersections, valves, pumps, and mixers all within the chip itself. This means we can split the original drop into even smaller droplets, sending each one down a different path for a unique test. We can mix a droplet with a specific chemical, called a reagent, to trigger a reaction that reveals the presence of a certain molecule. We can even use tiny filters to separate different components of the blood, like separating the red blood cells from the plasma, all automatically and all within the confines of this minuscule device. It's a beautifully orchestrated dance of fluids on a microscopic stage. This level of control is something you could never achieve with traditional lab equipment. In a big test tube, you're working with milliliters of blood, and mixing is done by shaking or spinning. But on a microfluidic chip, you are working with nanoliters. That's 1/1000th of a liter. At this tiny scale, fluids behave differently. The flow is smooth and predictable, without the turbulence you'd see in a larger volume. This allows for incredibly precise reactions. It's the difference between trying to paint a detailed portrait with a giant paint roller versus using the finest tipped brush. Microfluidics gives scientists that fine-tipped brush, enabling them to perform complex multi-step procedures on a sample size that was previously thought to be completely unworkable. Ultimately, microfluidics is the foundational technology that makes single-drop analysis possible. It solves the first major problem: how to handle and process a liquid sample that is thousands of times smaller than what traditional labs are used to. By creating these tiny automated plumbing systems, scientists can ensure that the precious drop of blood is used as efficiently as possible, with not a single cell or molecule wasted. It lays the groundwork for the next revolutionary step: building an entire laboratory that can fit onto that very same chip, turning these simple, fluidic highways into a powerful diagnostic processing plant that can fit in the palm of your hand. This is where the lab on a chip comes into play. Now that we have these tiny fluid highways, the next step is to build the rest of the laboratory around them. Diagram, functional zones on a lab on a chip. This creates what we call a lab on a chip. It's not just about moving the blood around, it's about performing all the functions of a full-scale lab: sample preparation, analysis, detection, all on one small integrated device. Imagine taking all the big clunky machines from a clinical lab, centrifuges, analyzers, incubators, and shrinking each one down to microscopic components embedded on the chip. That is the ambitious goal of lab-on-a-chip technology. Let's follow a drop of blood on its journey, a single drop falling toward a chip inlet. The blood enters the chip and goes through a preparation stage. Microfilters separating cells from plasma, acoustic channels sorting cells by size. The goal: get the sample ready for analysis. Next, the sample is sent into different reaction chambers. Chambers preloaded with reagents to detect specific biomarkers. One chamber, antibodies binding to a virus. Another chamber, chemicals reacting with glucose. Dozens of chambers, like many test tubes running in parallel. After reactions, the final crucial step: detection. Tiny optical sensors detecting color changes, tiny optical sensors detecting fluorescence, glow under LED, tiny electrical sensors measuring current or resistance as molecules bind. Each detector sends a signal to an external reader or a computer for processing. The chip translates biological events into simple digital signals, a system that is incredibly fast and efficient. From hours or days to a matter of minutes, automated closed system, lower risk of human error or contamination, transforms diagnostics into quick, cheap point-of-care testing. Power moves out of the lab and into the hands of doctors, nurses, eventually even us at home. So we have this amazing lab-on-a-chip technology that can process a tiny drop of blood. But what exactly are we looking for inside that drop? We're searching for biomarkers. A biomarker is a measurable indicator of some biological state or condition. Think of it as a clue. Cholesterol is a biomarker for heart disease risk. Glucose is a biomarker for diabetes. The presence of specific antibodies can be a biomarker for an infection. Your blood is teeming with thousands of these biomarkers: proteins, hormones, DNA fragments, cells, and other molecules, each telling a part of the story of your health. The challenge is that some of these clues are incredibly faint. In a large vial of blood, you have a relatively large amount of each biomarker, making it easier to detect. But shrink that sample to a single drop, and concentrations become minuscule. This is especially true for detecting diseases very early. For example, a tiny tumor sheds only minute cancer-specific DNA or proteins into the bloodstream. Finding these few molecules in a single drop is like finding one grain of sand on an entire beach. Traditional detection methods aren't sensitive enough to pick up that weak signal. This is the needle in a haystack problem, the biggest hurdle for single-drop diagnostics. It's not enough to just build a small device, it must be extraordinarily sensitive. Scientists design reagents and sensors so specific they bind only to the target molecule, ignoring millions of others. This requires deep molecular biology and chemistry. It involves creating custom-designed antibodies and DNA probes that act like molecular magnets, attracting and capturing their specific target even in vanishingly small quantities. A single biomarker rarely tells the whole story. Complex diseases create subtle patterns across many biomarkers at once. One protein might go up slightly, another might go down, a third might appear slightly altered. The real diagnostic signal is in the combination of all of them. A successful single-drop test must measure a whole panel of faint signals simultaneously, which makes the science far more complicated. Beyond the challenge of finding rare biomarkers, there is a more fundamental problem: consistency. Finger prick drops are not the same as venous blood. Venous blood is well mixed and stable. Finger prick is a messy mixture: capillary blood, tissue fluid, skin cells. Composition can change from one prick to the next, or from the first drop to the second. Variability ruins diagnostics. Measure cholesterol. First drop diluted by tissue fluid, cholesterol reads falsely low. Second drop, different red cell to plasma ratio, reads swing the other way. Medical tests must be reliable and reproducible. Achieving that precision with finger pricks is extremely hard. Systems must account for these variations. Contamination is the other big problem. The trip from finger to chip is risky. Skin isn't perfectly sterile even after wiping. Dust, lint, skin oils can enter the drop. In a vial, they're diluted. In a single drop, they matter. A speck can block a microfluidic channel or ruin chemical reactions. That causes failed or inaccurate tests. Early single-drop attempts, most famously Theranos, failed because of these issues. They underestimated how hard clean, reliable sampling is. Modern fixes, smarter collection methods, chips with onboard sample purification, and sophisticated algorithms to detect and correct errors. The science is as much about handling messy samples as it is about the chip.